Although seed treatment can reduce the number of pathogens present in or on seed, it cannot guarantee total elimination of pathogens. Under the warm, wet conditions of sprout growth, bacterial pathogens can proliferate. Even if only a few pathogenic cells survive seed treatment, they can grow to large numbers during sprouting and contaminate the production batch. Therefore, testing spent sprout irrigation water or in-process sprouts is an additional hurdle to help ensure that pathogens are not present before sprouts enter the food supply. According to the produce safety rule, spent sprout irrigation water means water that has been used in the growing of sprouts. Sprout growers are required to test spent sprout irrigation water or in-process sprouts from each production batch of sprouts for E. coli 0157H7 and Salmonella. In this video, we will demonstrate the key steps involved in sampling of spent sprout irrigation water and in-process sprouts. A written sampling plan is required by the Produce Safety Rule and should include step-by-step procedures of when, how to sample, the specific test method, and the volume of spent sprout irrigation water or sprouts to be collected. The sampling plan must include the sample type, spent sprout irrigation water or in-process sprouts, and the number and location of samples to be collected for each production batch of sprouts to ensure that collected samples are representative of the production batch. The written sampling plan must also include a corrective action plan for responding to positive test results. Sprout growers must sample and test spent sprout irrigation water or sprouts from each production batch of sprouts. A production batch of sprouts is defined as all sprouts that are started at the same time in a single growing unit, whether or not the sprouts are grown from a single lot of seed. A single growing unit could be a single drum or bin, or a single rack of trays that are connected to each other. The optimal time for sample collection is when pathogen levels are likely to be at their highest to maximize the likelihood of detecting pathogens. This time may vary depending upon the type of sprouts or sprouting practices. Research conducted on alfalfa sprouts showed that pathogen levels peak approximately 48 hours from the start of sprouting, as illustrated on this growth curve. It is recommended that spent sprout irrigation water or in-process sprout samples are collected as close to 48 hours from the start of sprouting as practical. For seeds that are pre-soaked prior to sprouting, the pre-soaked time may be included in the calculations to determine the timing of sample collection. The Produce Safety Rule requires that samples of spent sprout irrigation water or sprouts must be collected aseptically. It is important to train sampling personnel on aseptic techniques to ensure that the sample collection process does not accidentally result in the contamination of an otherwise clean sample. Sampling personnel should wear clean garments and wash hands before putting on sterile, preferably single-use gloves. Containers, equipment, and tools used for sample collection must be sterile in order to collect samples aseptically. If reusable tools or sample containers are used, they should be sterilized by heat at 284 degrees Fahrenheit or 140 degrees Celsius for 3 hours in a dry heat oven. The Produce Safety Rule requires that the collected samples are representative of the entire production batch of sprouts when testing for contamination. Therefore, during sample collection, efforts should be made to cover as much as possible the production batch of sprouts. For example, collecting spent sprout irrigation water that passes through the entire production batch, or collecting in-process sprouts from different physical locations across the production batch. It's important that sampling personnel be trained on how to collect a representative sample in accordance with the written sampling plan. When sampling spent sprout irrigation water from a rotary drum, the steps may include wash hands and put on a pair of new gloves. Open the sampling container. Hold the lid with one hand and the container in the other. Do not place the lid on a counter. Immediately collect 1.5 liters of water from a drainage point of the drum. Close the container and seal it immediately after collecting the sample. Make sure that each sterile container has been labeled. When sampling spent sprout irrigation water from a bin where mung bean sprouts are grown, the steps may include Adjust irrigation water flow to an appropriate rate. For example, 1 gallon per minute prior to sample collection. Wash hands and put on a pair of new gloves. Open a sampling container. Collect a representative spent sprout irrigation water sample from the bottom of the growing bin. This may be performed by attaching the end of a long stick to the collection bottle and sweeping the collection bottle or tray across the entire bottom of the growing bin. Close the container immediately and put it in a cooler, making sure that each sterile container has been labeled. Although testing spent sprout irrigation water is preferred over testing sprouts, in cases where sampling spent sprout irrigation water is not practical, for example, sprouts that use very little water, sprout growers must test each production batch of sprouts at the in-process stage. The principles that apply to sampling spent sprout irrigation water, such as aseptic techniques, proper sampling time, and definition of the production batch of sprouts also apply to sampling in-process sprouts. To ensure collected samples represent the production batch, multiple sprout samples must be collected from multiple locations within the growing unit. For example, from top to bottom, side to side, and front to back of the growing unit. It is recommended that at least 30 subsamples, approximately 50 grams each, be aseptically collected. The total sprout sample will be at least 1,500 grams, or 3.3 pounds from each production batch. Sprout subsamples should be kept in a cooler before being sent to a lab for testing. Collected spent sprout irrigation water, or sprout samples, should be tested by a qualified testing lab. Prior to conducting in-house testing or sending the samples to a laboratory for testing, sprout growers should check to verify that the samples are clearly identified with the production batch number and other identifying information for the sprouts and the sprouting operation. The microorganisms for testing should be specified on sample submission forms. Testing methods and other testing-related information should be clearly communicated to the lab via phone calls or emails. If using an external laboratory for testing, sprout growers should verify that the methods used by the laboratory to analyze the collected spent sprout irrigation water or sprout samples are either the methods FDA referenced in the Produce Safety Rule, or alternate methods that are scientifically valid and at least equivalent to the FDA reference methods in accuracy, precision, and sensitivity for detection of E. coli, O157H7, or Salmonella. When the initial screening test returns negative results for all pathogens tested, product can be released. In order to comply with the Produce Safety Rule, when the results from the initial screening test for either E. coli, O157H7, or Salmonella is a presumptive positive, further confirmation is required to verify that the pathogen of concern is truly present. Confirmation of a presumptive positive result from the screening test must be conducted on the same enrichment sample that resulted in the presumptive positive result. Additional testing of spent sprout irrigation water sample collected from the same sprout batch is not a confirmatory step. Additional testing of the leftover from the original non-enriched spent sprout irrigation water sample that had a presumptive result is also not a confirmatory step. If the confirmatory testing returns a negative result, the products can be released. But if the confirmatory results were positive, the products need to be discarded and initiate corrective actions, including reporting positive test findings to the firm where seeds were purchased, such as the seed supplier. Keep in mind that the production batch of sprouts must not enter commerce unless the results of the test are negative. Operations may consider establishing and implementing a hold and release program to ensure all products are held until results are received from the laboratory and released, or subjected to the corrective action plan. Procedures for a hold and release program may include: 1. Establish a lab sample tracking and product release sheet. 2. Record relevant information such as sprout type, production batch number, date and time of collection, pathogen targeted for the test, and so on the sheet for all the samples shipped to the lab. 3. Upon receiving test results, match the production batch numbers indicated on the test result form with the ones on the lab sample tracking and product release sheet. 4. If the test results are negative, initial each negative result in the lab sample and tracking product release sheet and record the date. 5. Responsible person must initial and date the completed lab sample tracking and product release sheet to verify the product being shipped has been cleared. If there's any positive test result for Salmonella, E. coli, O157H7, or any other tested pathogens, put a hold on the products and contact supervisors immediately. Verification of the hold and release program needs to be conducted on a regular basis. Each operation must have a written corrective action plan in place so that if a positive test result does occur, corrective actions could be taken quickly. As specified in the Produce Safety Rule, a number of corrective actions must be taken. These include: discard any sprout production batch that tested positive for pathogens, clean and sanitize anything that came into contact with a contaminated production batch or its spent sprout irrigation water. For example, drums, trays, bins, buckets, tools, and other sprouting equipment, and food contact surfaces in surrounding areas such as packing tables, floors, drains, and walls. Discard the seed lot used to produce the contaminated sprouts or return it to the supplier. Notify the seed supplier about positive sample findings associated with their seed lot. More details of corrective actions can be found in the Sprout Safety Alliance Training Manual. Sprout growers are required to establish and maintain records related to sampling and testing of spent sprout irrigation water or in-process sprouts. The required records include a written sampling plan, including the corrective action plan, records of any analytical methods used, results of all required tests conducted by a third party or in-house laboratory, an adequate description of the sprouts applicable to the record, for example, production batch number and seed lot number, and location of the growing area from which the sample was collected, and records of corrective actions taken. Please note, all records must be signed and dated by the operators at the time when the tasks were performed, and reviewed by a supervisor or other responsible person. These records should be kept for 2 years. In summary, testing spent sprout irrigation water or in-process sprout is required by the Produce Safety Rule. It helps to prevent contaminated sprouts from entering commerce. It's also a verification step to ensure prior control procedures are effective.